So good afternoon, everyone. If you're based in the UK, Europe, and have a good day if you're joining from elsewhere in the world. So we are delighted to run the second webinar from the UK Centre of Excellence in In-Silico Regulatory Science and Innovation that is funded by Biomet UK and the Medical Research Council in the UK with collaboration with the MHRA, which is our regulatory agency. And we have the great opportunity today to have colleagues who are going to talk very soon and given a number of examples. What I want to very briefly explain what the Centre of In-Silico Regulatory Science and Innovation is, but it's a recent initiative from the UK government with the objective of reinforcing the regulatory capability in the UK. So we have 7 centres that have been funded. They're not funded based on a particular institution, but they're national networks that are led by one institution, in this case, the University of Manchester, but otherwise engage expertise across the UK and beyond, and with the emphasis in some particular topics. So ours is the use of in-silico technologies as part of the life cycle of R&D of medical product innovation, and also for generating regulatory evidence. And this is a really important initiative. We have 85 organizations that are participating, ranging from academia, industry, regulatory bodies and other government agencies, and finally as well participation from the public and patients as well as collaborators from the NHS and various large infrastructures like UK Biobank and the various NHS organizations. So without further ado, I'd like to introduce you to Stephan Kreuzer, who is going to be convening the rest of the webinar, and he will introduce the rest of the event. Thank you.
All right, thanks, Alex. Yeah, so pleasure to be with you guys here today. And as you see, we have Philippe Favre and Stephan Schaller here to speak with us alongside myself as case studies relating to the use of in-silico tools. Really excited today to share some examples. I'll talk about one from the cardiovascular space. Stephan will talk more on the PKPB and QSP pharmacological intervention side of things, which I'm really excited to hear about because I don't know very much about it. So that'll be great. And Philippe's going to share his experience at Zimmer in the orthopedic side of things. So should be a really nice discussion here. Please share your questions in the chat in case we have an opportunity at the end of today's discussion to go through those questions. But without further ado, I'll go ahead and share my screen and we can get going on the webinar. Let's get this. All right. Yeah. So this initial conversation that we're going to have relates to the enrichment in silico clinical trial. So this is an effort, as we'll talk about in a little bit, coming out of the States with involvement worldwide, where we went through the process of really going through an in-silico clinical trial and trying to understand what some of the challenges would be, what some of the necessary frameworks might look like, how to assess credibility, things of that nature. So it's a great experience, and I'm happy to share some examples of that. We'll talk a bit about the specific project, but what I also want to do here, particularly in relation to what Alex discussed for the CEiRSI in general, describe how this particular enrichment project might fit into the framing of the pilot projects for the CEiRSI project itself as a whole. So we'll talk a little bit about that, and I'll describe how, if we were doing the enrichment project as a CEiRSI pilot, we might approach some of the initial work. So that'll be the framing of things. So we'll go through a bit of an introduction. We'll talk a bit about credibility, some things that many of you have heard before, but just to set the baseline for what we'll talk about later. I'll go into a brief background for the enrichment project itself, talk a little bit about what we meant with that project, what we did, and so forth, and then really go into that as a case study, both as a case study for in-silico clinical trials and examples of how industry and regulatory agencies are thinking about in silico clinical trials, as well as a case study in how to put together the framework and the discussion to utilize some of those credibility frameworks that might be helpful for some of the later CEiRSI pilot projects. Now, as it relates to introduction, so here's myself. I'm a senior managing engineer with a company called Exponent. Went through a few schools in the States here, primarily focused on modeling and simulation, as well as testing. One of the things that we like to do, and obviously we need to do quite a bit of, is testing and analysis in order to establish credibility. So I pride myself in doing both the modeling side of things as well as the testing side. Some publications on the right there that I was involved in. The bottom one in particular is one that we'll talk a little bit more about. That's the primary outcome of the enrichment project itself. Before getting into some more details on that and credibility frameworks, I do want to give a brief background on Exponent. And the reason why I want to do this is to underscore the emphasis on credibility assessments and the various factors and evidence that are necessary for credibility assessment. As a company, as we'll see, we do a lot of that work. But just to provide some scope in terms of where we're located, you'll see here the majority of our offices are in North America. I happen to be outside the Boston area. We have a number of offices in the UK and Europe, as well as some presence in Asia. So we do get involved across the world in various activities, engineering, ChemReg, a number of different regulated spaces. And a lot of our work spans in those regulated spaces, both in pharmaceuticals as well as devices. Again, this is really to point out the importance that we place on credibility evidence and generating that evidence. As you'll see here, these are all the various what we call practices, basically departments. Not super useful for much of this discussion, except to emphasize that when we're looking at a problem, just like everybody else should be as we're thinking about in silico trials, when we're looking at a problem, we really need to make sure that we're leveraging our expertise and our colleagues, whether they're within our institution or outside our institution, who are interdisciplinary in their background, who have different expertise in different complementary tools. So as I hope we're all familiar with and comfortable with, in silico trials, even though we talk about them on the computer, we, of course, need to be able to ground that in testing, in development of credibility evidence, preclinical stuff, post-market surveillance, all that sort of work. So when we think about our life sciences, a lot of our perspective and the perspective that I come at things with, and hopefully comes through as we talk about this particular example, is grounded in the various stages of the total product life cycle. For those who are not exposed outside of the simulation space, I would highly recommend that you take an opportunity to learn as much as you can about some of these other areas that are beyond the experiment or the simulation side of things, because they do inform so much of what we do, as we'll talk about with respect to credibility. You know, some of those things, and I threw a whole bunch of pictures on this slide, but some of those things relate to things that are near and dear to my heart, like fatigue testing and mechanical assessments, tribology, corrosion, those sorts of things. But they really do span across the total product life cycle. And I think that's super important to understand, you know, regardless of Exponent, but it's really important to think about, because when you are thinking about establishing credibility for an in silico trial, we need to make sure, particularly in devices that we're going out, we're doing that testing. It's also true in pharmaceuticals and biotech. You need to go out and you need to do the various assessments that are going to complement your simulation approaches. So I pride myself in being involved in that on a regular basis. I walk down into the labs and see the guys and the gals doing the work there. Super important to see it across both simulation as well as testing. That said, when we start to think about credibility of modeling and simulation, I'm going to zoom a little bit out here, and I'm going to touch on some of the important aspects that I see are really valuable for as it relates to the CEiRSI effort in general. Some of the things that are incredibly important for the evolution of in silico trials, for the adoption of in silico trials, are the things that Alex and the team are trying to do. So everybody that's in this call, everybody who's involved, are engaged in this incredibly important effort. And I've highlighted a couple of things here that are key points on the CEiRSI effort. The main one that you see there is a focus on applying and evaluating the strengths and limitations of existing international credibility frameworks. So one thing that's really valuable here is just taking these pilot projects or whatever project we're thinking about and putting it through these frameworks so that we all get experience with and understand the limitations, the strengths, the ways in which we can do these things, and the benefits of using these frameworks. We'll see as I walk through the example of the enrichment project from a credibility standpoint, the application of at least one framework to that project of the enrichment project. And we'll see some examples of that. I'll point you to some literature you can see elsewhere. But I just really want to emphasize the importance and the value of this group getting together, particularly those in the audience who are listening to this who are more from the regulatory side of things. The benefit of talking amongst the technical folks and the regulatory folks is tremendous. Quite frankly, we can't move these technologies forward until we have these conversations. And everybody being in the room for this webinar, for the pilot projects, for everything that's coming out of the CEiRSI is going to be tremendously valuable, obviously for our own institutions and our own careers, but also for public health in general. So count me as a fanboy of the CEiRSI, of course, but I think it's really important that we're all doing this thing. Now, as we think about frameworks, the one I'm going to leverage the most in my work here and my discussion is this FDA guidance on credibility and modeling simulation. So as you see there with the dates, this came out about 1.5 years ago. The draft guidance came out even before that, of course. And it's a discussion that goes through much of what would be necessary to assess the credibility of modeling and simulation, as the name implies. I think it's accurately named. It involves a lot of discussion. It leverages some other standards, most notably the ASME V&V40 standard that many have heard about and Jeff and team discussed last webinar. But it primarily establishes a workflow and a framework that allow you to think about your problem through this lens of assessing credibility. And so whatever international framework you're looking at is probably going to be established in a similar way. The bits and pieces might be a little bit different. It might not look as complicated as this, but it's going to include many of these same components. I want to point out this first figure that's in the guidance on credibility, or assessment rather. There's a number of different steps in here, and these steps are incredibly important for thinking through the problem. One huge caveat that's always discussed with these types of conversations is that the framework does not tell you how to do credibility assessment. It puts a structure together that walks you through that process. It's up to you and the team that you've put together to walk through that process, to address the various issues that are raised, to use your engineering judgment, your regulatory judgment, your assessments to be able to answer some of the questions. But it does put the skeleton or the workflow in place. And I think that's really helpful because we're going to use it today. And I suspect that many of the pilot projects and many of the efforts that you guys are doing outside the CEiRSI need to leverage this as well. So the prior slide showed the framework in all its gory detail in that figure 1. I have sort of extracted it here into various steps. And we're going to step through these for the enrichment project. And I'm going to use this particular framework, again, to think about how one might start the process of credibility assessment in much the same way that the pilot projects are going to do. So we'll see this again in a second here. And I'm pointing this out specifically because if you look at the timeline that Alex put together and describes in the launch webinar, this is taken right from that. There's a couple of things. One, we are in the stage of the awareness webinars, which is the blue arrow on the left there. But there's all of these pilots that are aligned here across the course of the year with various timings, and the more details on that are elsewhere. But the basic idea with these little green triangles is that we have these pilot panel sessions. Now, this is my interpretation of things, but my understanding of these pilot panel sessions is for each of these pilot teams to have worked through some of the details that I'm going to describe for the enrichment project, to be able to then present some content to a larger group, to be able to talk through some of the challenges associated with whatever that pilot is. Now, when we go back to this guidance and we line this up, The way I'm thinking about it, at least, and I might be wrong on this, but the way I'm thinking about it is that these pilot panels are essentially going to walk through these first 6 steps of, now it doesn't necessarily have to be this framework, but a framework like this to address the questions that are in this red box. We'll talk about what each of these steps are, and of course, we'll use the enrichment project as an example to walk through these steps to give some concept and hopefully a concrete example of going through this process. One quick note on this, everything that's in the red box here is essentially prospective. The way the framework lays things out is it asks you to walk through many of these questions and address them and put them into a document and then perform a prospective adequacy assessment. Talk a bit more about that, but basically, you want to look holistically at what you put together and decide whether it's going to be adequate. In the US, this is a stage where you might go to the FDA and you might have what's called a pre-submission meeting to talk through your plans for how you intend to establish credibility. And then step 7, assuming you get the go-ahead and everybody's on the same page, step 7 is going and gathering that evidence. So what we'll talk about, and what I think is relevant from a pilot panel perspective, is everything that's up to that step 6 before obviously we go and we gather any evidence. And then once you gather that evidence, you can go and you can look at it in the post-study and you can think about whether your evidence, or your model rather, met the goals of your credibility assessment. So let's put that into context here. I'm going to do it through this enrichment in silico clinical trial project. It is a project, as I'll describe in a second here, that has quite a bit of history, about 5 years or so at this point. And we, from more or less wrapped it up last summer, and we'll talk about that. But it's an effort that came out of FDA and Dassault Systems to, again, basically just describe and try to figure out what the heck we're doing with these in silico clinical trials. And from that standpoint, I think it was successful, and hopefully we learned some stuff through that. So this is a title slide from that. Again, as I mentioned, it's a project with the FDA and Dassault Systems. A number of other folks were involved. This is an image of the folks who were at one point at least part of the project leadership, and some of this shifted around at various times. You see some folks who were sponsors, some PIs, some councils, and so forth. I put this slide up primarily, and I like discussing this because there's a lot of different people involved in this. As with the CEiRSI projects, there's a number of different institutions, but critically, there's also a number of different perspectives involved here. So you see just in the advisory councils, the folks who are engaged include clinicians, scientists, or general subject matter experts in various areas, as well as industry participation. You see the PIs include folks from FDA, primarily OSL, as well as somebody who leads the Living Heart Project and some virtual human modeling work at Dassault Systems, Steve Levine. So this group had a wide range of perspectives. It also included academics. So there are a number of folks involved, particularly in the nuts and bolts of what we did, who were asking really fundamental questions and gathering that expertise, and we'll show their names in a little bit here. But this project was geared towards mitral regurgitation. So there was a little bit of background in terms of why we chose that or why it was decided. But the basic idea, and most of you are probably familiar or at least can piece together the idea based on the name. But mitral regurgitation is a misfunctioning or dysfunction or whatever you want to call it for the mitral valve connecting the left ventricle with the left aorta, or left atrium rather. It's basically flow that goes in the wrong direction, and it's a very common problem. And there's a lot of details involved in it, of course, otherwise it wouldn't be worth studying. But there are fortunately a couple of different devices that are out specifically to treat mitral regurgitation through what's called an edge-to-edge repair technique. There are a number of other devices in development for regurgitation that might be replacement devices for the valve. But just to ground things, and you'll get a better appreciation for this hopefully as we continue, but just to ground things, the idea with this particular device is that you basically clip together the leaflets that are not sealing properly. And through that device, clipping those leaflets together, you reduce that level of regurgitation, and that has all sorts of downstream benefits. So, there's tons of details in that, but the basic idea is, can we simulate those devices in a regurgitation scenario? It was geared towards reproducing something called the COAPT trial. This is the primary output paper of that trial. It came out, I don't even remember the year, maybe 2018. And you see the Kaplan-Meier plot on the right-hand side there, very compelling, not uncontroversial for those who are familiar. There's quite a bit of discussion about this particular trial, as well as some other trials with different patient populations and so forth. I mention all of this, though, just to point out that as we were doing the enrichment project, it wasn't really a prospective project in the sense that we had nothing to compare to. The leaders of the project and the organizers specifically chose something that we had some experience with generally as a community. So the trial had wrapped up by the time we started this. There was information to gain, but we were starting from a point where we basically knew the answer. And we were walking through the process of running the In-Silico clinical trial with that answer in hand, which obviously has some advantages to it. But that was the goal. And what we ended up coming out with from this project is a lot of fun times and good adventures and so forth, but also this particular paper shown on the left-hand side that discusses the observations, quite frankly, that we had. What's interesting about that paper, and I'd encourage anybody interested to look it up. It is open access, so that's awesome. But what's interesting about it is it doesn't contain the technical details. There's a number of papers, there's probably a handful of papers that sort of fit within this vein of describing frameworks and talking about some of the challenges and so forth without actually putting down the case study results and the details. That work was left for the appendix. Basically, we wrote the paper and various folks told us to take it and put it in the appendix because they wanted the main document to be relatively standalone. I do want to emphasize that because if you are interested in what we did and you want to learn more, there's quite a bit of really good detail in the appendix, and it's technical detail. So it's very interesting if you're coming at it from a technical standpoint. So don't just download the paper, although that paper is very good. Make sure you also take a look at the appendix and dig into the details there. And I'll share a lot of those details, of course, right now. So let's take this project and let's throw it into this credibility framework. In particular, let's put this project in the framework, as I mentioned, that I think is relevant for the pilots for the CEiRSI project, namely walking through the initial steps of at least the credibility guidance from FDA, in order to establish some of the details of the approach as if we hadn't done it yet. So I'm kind of going to wind back the clock a little bit. You're going to see some images that obviously point out that we had done it, and I'm going to talk about what we did. But I want to have a perspective of how we might have addressed these questions up front if we had used the framework prior to starting the project. The framework did come out after we started the project, and I think our work on the project heavily informed the development of the guidance, because many of the folks who were involved in writing that guidance were also on the project. So it's a little bit of a test case there. So it's a unique point in time and project to talk through this. But the main aspects of this particular framework that we'll describe here are what's shown on the screen. And for those who aren't familiar with some of these terms, some of them were discussed by Jeff and team last time. But for those who aren't familiar, I'll talk about each of these various terms from the perspective of how the framework and the guidance define them. But we'll discuss each of these major steps, and I'll use the enrichment project as the example for addressing each of these steps in the process. We will not talk about the actual evidence or anything that relates to the results of the ISCT. That's something that I'd be happy to discuss in a different framework or a different place, and you can also find in the appendix. But the first step of all of this is to have identified the device of interest and to think a little bit about that device and what you want to tackle. What we'll see in the next slide with the question of interest really does require you, of course, to have a device in mind. Certainly in most circumstances that are practical, you're going to have a device in mind already. Because you're working at a company and the company is developing a device, or you're working on behalf of that company, whatever. But as mentioned, our device of interest is a clip device, an edge-to-edge repair clip device. In particular, we took most of our inspiration from the Abbott MitraClip. There is a competing device from Edwards Life Sciences called the Pascal that's at least from 30,000-foot view, very similar. It's an edge-to-edge repair device. But I do have that red box on the bottom there because the indication of interest that we're focused on, it's not only the device of interest that matters, it's of course also the patient population we care about. In this case, the patient population are those with secondary MR. So this is essentially your left ventricle has gotten too large, and as a result, the leaflets don't touch each other anymore. Some more details in it, probably a gross simplification, but that's essentially where we're at. So that's a device of interest. And once you have that nailed down, and of course the pilot projects all came into being, or at this stage at least, have a device of interest and a stated goal. Now the next step, and some of this, I apologize, is going to be a little bit wordy, but the next step is to establish a question of interest. And this is on the screen here. We have an example of the question of interest that we tackled, and also the definition at the top that's taken from the FDA credibility guidance. Now, it's really important to think about with the question of interest, and I cannot emphasize this enough. This question of interest is not solely what's being done with the modeling. It's not solely what's being done with the In-Silico trial. It is a much larger question that is at least partially going to be addressed with the computational model. But as it says here in the definition at the top, potentially other sources of information. Most likely there is a lot of other information coming into play. But the question of interest is you can think of it as a large program scale question. And for us, for the enrichment project, it's interesting because our question of interest was around enrollment criteria for an investigational device exemption trial. So we were basically, as the language describes there, we were looking at whether we can use modeling and simulation to refine or enrich the enrollment criteria for a trial. So the question of interest here was, what should that enrollment criteria be? So notice in this question of interest, nowhere are we talking about the model itself. Instead, this is the large scale question that we're trying to address with the help of the model, but with a whole lot of other information. Now, that's the question of interest. The next thing we have to dig into is the context of use. This is a lot wordier, but the context of use is a follow-on from the question of interest, and it's a detailed description of how whatever you're modeling is going to be used to answer that question of interest. A lot of text here. I highlighted some of it so I could remember what to focus on, but when we're thinking about the context of use for the enrichment project, we are using it to predict, as it says here, the acute post-operative mean left atrial pressure in a virtual patient cohort in order to predict mortality so that those results can inform potential revisions to the basic baseline enrollment criteria. So what I like about this context of use is it's very specific. Now, we can make it very specific because we essentially wrote the context of use after we ran the trial. So a little bit of a cheating effort there. However, it's very specific. We talk about a specific outcome here, a left atrial pressure, mean left atrial pressure. We talk about how we're going to use that to predict mortality and how, again, that's going to be used to prospectively exclude patients who might not respond favorably to the intervention. So this is obviously the ISCT being geared for enrollment criteria. But that's the establishment of the context of use. And that context of use then goes, of course, to help answer the question of interest. Now, once we've done that and we've established both the question of interest and the context of use, we are at a point where we need to establish the model risk. Now, model risk is composed of two things. And the model risk is associated, as you see there with the definition on the top, with using the model to address that specific question of interest. And it's essentially around what happens if you get the model wrong? How does that address the question, or how does that impact, rather, the question of interest? The two components that go into the model risk assessment is model influence. So it's essentially how much are you trusting, how much of the ultimate information that you're going to use to answer the question is reliant on the model. What else are you bringing to the table? If it's just going to be the model influence is pretty high. If you have a whole lot of other information and you're just using the model to check something, model influence can be pretty low. And you see there the model influence that we've established for the enrichment project as a low medium model influence. So this is a qualitative gradation of model influence, and we'll see how that maps onto the model risk in a second. But essentially there's a moderate influence with the ISCT outputs, and the key in that second sentence of the boxed box is that the evidence is weighted less heavily than some other evidence. So we're on the low medium side of things. We're going to pay attention to the model, but it's not our primary source of information for establishing the enrollment criteria. You can see all the way down at the bottom, if we had a high model influence, where the model would have a dominant influence on the decision, there's no other information being brought to bear, and the flip side of it on the top if it was none. But we're in a situation where we're looking at a low medium scenario. Now, the second thing that we got to look at is something called decision consequence. So decision consequence, I'm going to show two of these graphs in these, or plots or whatever. They look very, very similar. The key point here with decision consequence is it's composed of, or at least one way to look at it, and the way we were looking at it, is that your decision consequence is composed of two components. One is the severity of harm. So if you get this wrong, how much is somebody going to be hurt? So that's the y-axis here. Now for class 3 device, as we classify them in the US, for a tier device, a mitral edge-to-edge repair device, if that device fails, it could have absolutely critical consequences. Not necessarily fatal, but it could very well be. Now, the probability of occurrence of harm relates to how often the device could have an issue. This particular scenario, we called that a probability occurrence of harm. And that's just based on some historical data that we were able to leverage. But it's essentially how often is something bad going to happen. But when you put those two things together, the way that we put this framework or made the decision for decision consequence, when you map those two, you get a medium high decision consequence. So it's a relatively important decision that's being made here, primarily because both the severity of harm and the probability are relatively high. So once we've established that decision consequence, now we can look at the second graph that looks very much like that. Like, this is colored the same way. We probably should color it blue or something else, so it's a little bit different. Regardless, once we take those two things, the model influence on the x-axis and the decision consequence on the y-axis, we can take our decisions that we made in terms of classifying those two things, put them in a framework or a matrix like this, and come out with what we established as our model risk. So in our case, we established the model risk as a medium model risk. So in the CEiRSI pilots, and as you're doing these frameworks, if you're doing them yourself, not in the context of CEiRSI, you've established your question of interest, your context of use, and you've addressed these two questions in order to establish what your model risk is. The importance of establishing your model risk is that it then sets the stage for the level of rigor that's going to be necessary for all of the other credibility assessment activities you're going to undergo. So when we look at that, we need to establish first the various models that we are thinking about. So once you have the models established, you can start establishing those credibility factors. Now, at this step 5 of this process, we're in a position where we need to say what we're going to do. So we've talked about our question of interest, we've talked about our decision consequence, all those sorts of things. But what is the modeling actually going to look like? Now, in our case, for this ISCT, we broke it into these submodels. And I'll talk about each one of them in a second here. But putting them all together is necessary in order to really run the full ISCT. Each of them are termed submodels because they all fit together in different ways to assess or to perform the full ISCT. They are not submodels in the sort of numerical finite element sense, although that could be included. They are more different ways of and different modeling techniques to establish some predicted performance of one aspect of an in-silico of a clinical trial, in our case, an in-silico clinical trial. The first one is the device. So very high level, the device that we're doing looks like this, or that we leveraged. It's very, very, very de-featured here. We were not looking at device safety or reliability necessarily in this project. Instead, we are just looking at efficacy. So all we needed to do for our model in this example, in this case study, all we needed to do was just make a clip that could clip in the same kinematic way that the MitraClip or the Pascal device might do it. And so those are the features that you see here. It's a very, for lack of a better word, it's a very dumb clip. It's just rigid plates and grippers and stuff, and they're just going to interact with the leaflets. So all very straightforward there. But for whatever your project is, for whatever the pilot studies are, or whatever, you're going to need to establish obviously what your device model looks like. And there might be multiple models depending on different sizing and so forth. We then went into establishing what a patient model might look like. There's a few different features here that I think are incredibly important. One is the ability to parameterize the anatomy. So in our case, we were not leveraging patient scans to create the anatomy. We were creating fully synthetic virtual patients. So we parameterized the anatomy, and then we're able to generate synthetic mitral valve apparati, which you see on the left-hand side there, with whatever parameters might be either measurable or conceivable based on the known anatomy of a mitral valve. The other thing that's shown on the right-hand side there, I think is not to be missed and is incredibly important, particularly in a cardiovascular sense, because it speaks to the systems biology aspects of these trials. So we didn't just have to model the valve. Because it's a cardio application, because there's feedback loops associated with how the body's going to respond to changing the performance of the valve, we also needed to include a hemodynamic model. And so what's shown on the right-hand side there in essentially a circuit diagram view is the hemodynamic model that is co-simulated with the finite element model. So we had to put all that stuff together in order to not only capture how the device was going to close the leaflets, but also capture how the body was going to respond. Then we're able to put the device into our virtual patients, and we see here just some screenshots of that process. So the device and the patient get in, or the device is placed, and it clips the leaflets together, and then you get to co-simulation, you get some orifice area, and you get some other technical details. But it's very, very important for something like this, and in our case, to understand how you were modeling the interaction between the device and the patient, particularly as it relates to the way in which a clinician is going to do it, as we see in a second here. So we need to have that flexibility to incorporate how a clinician might make decisions and implement an intervention, and the variability associated with that, because while clinicians are very, very good, they are not omnipotent, and they can, sometimes not achieve the desired results. And we need to be able to incorporate that. From a virtual cohort perspective, this is essentially saying we're going to throw together a whole bunch of these little models, and we're going to run a whole bunch of scenarios. Ton of details on this, but the basic idea is you need to be able to create a cohort that has the right characteristics relative to whatever your patient population is going to be. So some of the things we did here leverage machine learning. This was before ChatGPT, so it was before everybody was using AI for literally everything. But we did use AI, machine learning, whatever. And a lot of what we were doing here was focused on filtering to make sure that we had the right characteristics of our patients, which is an important focus that we'll talk about in a second. Next step is a clinician model. This is not particularly sexy, but what this talks about is basically incorporating the decision-making process of a clinician. For this particular indication, you could think about it as you have a valve, you have different places you could put the clip. A clinician is going to decide where to put that clip. They're guided by some information, but oftentimes they make a decision that's based on their experience. The way we incorporated this was we actually had a clinician in the loop, which we probably should have called it that because that sounds kind of cool. We had a clinician in the loop who was making some decisions about where to put various clips. So we showed them some preoperative simulations. They say, clip it here. We clipped it there. So there's a clinician making those decisions. In some circumstances, you don't have that benefit. We did. We took advantage of it. The last step here is a clinical outcome model. This is a model of how you're going to extrapolate your acute performance of your clip. Once you put that clip in, it's going to close some things down. It's going to change some stuff. How is that going to manifest in mortality or whatever your real clinical endpoints are? In our case, as the title of that implies, and as we talked about before, it was mean left atrial pressure. So the clip changed that left atrial pressure. We looked at that change and what the resulting value was after the procedure, and we used that to project out what the mortality might look like, whether it was good or bad. This is very hard to do. We had some benefit here because we were kind of recreating the wheel, if you will, because this trial had already been run, but that's a hard thing to establish. However, once you've established each of those models, you can then get into this fun business of establishing your credibility evidence and your credibility factors. I'm not going to go through details of every single model and what the evidence and factors are, but it's important to recognize that the credibility evidence is the evidence that you are gathering, whether that's computational, additional computational evidence, bench evidence, clinical evidence, whatever it might be, but the stuff that you're going to use to colloquially validate the model. It also includes verification, all this other stuff. But what are you going to do to establish your credibility? The factors for your credibility are related to the rigor with which you make your comparison. Now, the key point there, got in blue, is you need to establish credibility evidence that you're intending to gather, as well as the associated factors for each submodel, as well as the connections of the submodels, of course, that you're going to use in an ISCT. Now, the guidance does lay out various categories of evidence, and there's some details in there. Some of it is well-defined. Some of it leverages ASME V&V 40. Some of it you just kind of need to come up on your own with what the evidence is. But you see the various categories here, and it's useful to put them into the categories. But I want to emphasize that once you have a category, you then need to establish the various factors within each category. So it gets kind of spidery at this point because you have different pieces of evidence. Then each evidence has different credibility factors. What's shown in the main box here is taken from V&V 40, and it describes various factors associated with bench validation. You might have this for all the other evidence categories, but at least for bench validation, you have all these other things. Once you take them, the key point on the right-hand side there with those different numbers is that if you're going to compare a test to a simulation, you can make that comparison in a number of different ways. The rigor with which you make that comparison needs to be driven by your model risk. And so there's a whole bunch of discussion about that. But basically, you establish your evidence, you establish the factors you're going to use within that evidence, and then you establish your rigor within those comparisons for your comparison between your evidence and your model. And that rigor needs to be commensurate with your model risk. So I put a whole bunch of these on here to underscore the point that your credibility factors need to be established for every piece of evidence, for every one of your submodels that you have in your project. So a little bit of homework here. You got to put that together. It's a little bit of effort, but it does create a nice game plan and allow you to communicate with regulatory folks. It also emphasizes the rigor with which you are organizing your program. So putting all this stuff together and putting it in front of somebody really makes you look like you know what you're doing, hopefully, and hopefully you actually do know what you're doing. Once you get that evidence, and actually I should point this out, you can have different categories of evidence that are more relevant for different submodels. This is a breakdown here from the paper. You could take a look at it, but essentially the different categories of evidence are not relevant or applicable for all the submodels. And some of them you might be able to leverage, some of them you might not, but it's up to you and based on the situation, of course, what you're going to incorporate. Now, once you do all of that, you come to a very fluffy part of the process where you are thinking about the adequacy of your credibility evidence. So there's two stages of adequacy here in the guidance. There's prospective adequacy. So you're basically saying, I haven't gotten the evidence yet, but if I get it and it looks like I think it's going to look, is that going to be enough? And then there's the post-study adequacy assessment where you've commissioned the studies, you've gathered the evidence, you've thought about all the uncertainties, you've done your design of experiments and your validation of methods and so forth. And now you're looking at and you say, okay, I have these results. Are they actually supportive of my model? Do I think that my results and my credibility is adequate for whatever my question of interest and model risk are and so forth? Some various questions here, some really high-level ones include, are you incorporating testing or gathering of evidence for all relevant features of your model? What is the reliability and accuracy of the factors, the credibility factors that might not be traditional bench evidence? As a community, I think we're really good at comparing models to benchtop tests. We are somewhat less good at comparing models to in vivo results. So, what's the accuracy and reliability of that comparison? That's a little more poorly defined, I'll say. And lastly, in terms of big questions, what kind of clinical unknowns are there that you're just not capturing in your model, and what might be the potential effect of those clinical unknowns? The human body, of course, very complex. You're not going to include all of it in your model, and the things that you're not including, why do you believe that you're justified not including those? So those are some of the key questions to address as you're thinking about adequacy of your evidence. Now, the last stage here, and this isn't really part of the guidance, but I think this is incredibly important for the CEiRSI projects and the pilots, and that's a discussion of the practical feasibility of acquiring the credibility evidence. It's all well and good for you to go in a room with a group and come up with a million different things that you could measure, and to say you're going to do the world's largest DOE to understand all the sensitivities, and you're going to have all the equipment in the world available to you and all of these things. That's not very practical, of course. Budgetary constraints, not the least, constraining some of these efforts. So you have to be very practical with what you're actually going to gather. And in some circumstances, it's very clear whether you're going to be able to feasibly acquire the data. In other circumstances, that's not going to be the case. I highlighted here some select challenges that are associated with the enrichment project in particular. One is, the first one is just the mapping between the acute performance and clinical endpoints. The second is establishing patient-level data. So not just distributions of each metric, but correlations between metrics, things of that nature. And the third one is just the ability to predict on a patient-specific level performance. So each of these boxes likely could have other challenges, but I think it's critical for these pilots and for any effort that you're undergoing before you start trying to gather your evidence, super duper incredibly critical to think about the feasibility of gathering this evidence. You as an individual, as an organizer of your modeling, are not going to be credible in your approach when you go to business leaders and people who you're asking to fund these efforts if you haven't thought about the practicality of what you're trying to gather, and you haven't been honest with yourself and with the team in terms of whether you think you can actually get this stuff. So this step is not to be missed because your ability to convince people, quite frankly, is often most closely linked not to your skills in doing the modeling, but the practicality of gathering this evidence that's going to fit into your credibility. All right, so once you get all that stuff, you can then go and pitch it to whoever the folks are who might need to sign off on gathering the evidence, which can be a costly stage if you're not able to recycle some of your evidence. I'm going to pause there, stop there, primarily because I think that's where the CEiRSI projects, the panel discussions for the pilots stop. But as I stop there, I do want to throw up a whole bunch of folks for acknowledgements. We already saw the enrichment leadership team on the left-hand side there. Obviously incredibly important from a leadership perspective. I put the core modeling team from Dassault on the bottom right there. All of those individuals really did a tremendous amount of work to run the analyses and do lots of stuff that I didn't have a chance to talk about. If you happen to run into any of these individuals in the wild and maybe they're applying for a job, or maybe you have an opportunity to talk to them, do give them the time of day because they are excellent. And then in the top right-hand corner there, I have the folks who are named on the paper, some of whom are shown in the modeling group, but many others who were involved who don't have a picture up here, but who are incredibly important. All right, so I'm going to stop there. And like I said, happy to address questions outside of the context of this meeting and potentially after my very capable colleagues who are here to present have had a chance to discuss their work. So with that, I am going to kick it over to Stephan and allow him to take it away.
All right. Thanks, Stephen, for setting the stage for this topic. So I'll be switching gears a bit and going less on a high level kind of regulated or best practice type of overview, but move into more focus on modeling and simulation in the pharma industry, focusing on the development of models and their application and more or less evaluating this in the context of regulatory use. So if everything works, you should be seeing my screen now. So I'll be talking about PBPK and QSP models. So that's physiologically based pharmacokinetic and quantitative systems pharmacology. That's quite a mouthful, but these are very mechanistic, physiological type of models that are used in a translational context, so learning across biology. And as digital twins of humans or animals are used to evaluate both clinical or non-clinical outcomes and translation of these into the clinic. So somehow my slides are not moving. Oh, that was just a lag. Okay, so I'll give a brief intro, then talk a bit about the software that we're using as it's open source, so a bit of a special case. And then looking at these systems, pharmacology type of model platforms, discussing around the platforms and the challenges that we have around the qualification. So basically related to what Stephen just discussed in terms of the credibility. So how do we assess these platforms and their qualification for intended use, their verification, and how might we find software solutions that could help us in managing these aspects, and to then outline also a bit of a strategy of how such software solutions can help us do this in an efficient way, ensuring quality and credibility of such models, and then giving this a bit of a context in industrial use. So, well, I'm from esqLABS. I'm the managing director of esqLABS, and we are a contract research organization working in the broader life sciences. So we work across pharma industry, the chemical industry, or device industry, developing digital twins of human systems, as I mentioned earlier, to assess drug kinetics and effect. We have a team dedicated to pharma, one dedicated to the chemical industry, and then a software team. We work with large and small industry across the scope, and we are quite heavily involved in public research funding, and are funded here across Europe from different initiatives and also government grants. And I very briefly want to give you a bit of a scope of where we work in. So as mentioned, in the pharma industry, which is called model-informed drug development, using these models to de-risk development steps of novel modalities. But then in the chemical industry, this application is called next generation risk assessment. So we're looking to assess exposure of animals or humans to pathogens or to chemicals that are potentially harmful, assessing the levels of exposure, and from that derive potential risk by also modeling potential effects or adverse effects or toxicological effects. And then the software development that we do and the software solutions we're trying to build. So in the model-informed drug development space for pharma, we work across the space, not to go into detail, but for example, looking into formulation-related aspects. So how do tablets, for example, behave when they dissolve? And how do I have to manufacture my drug and make it available so that it can be absorbed and is available for action in a organism or mammalian system? Then derived from that, of course, investigating that across species and then finding the ultimate perfect first in human dose. So looking really at exposure and de-risking the first clinical trial. From that, we can move on to investigate special populations, pediatric populations, pregnancy populations, or also disease type of populations, how this might affect drug exposure. Then a related case here might be the drug interaction, so polypharmacy, taking multiple drugs, and how that might affect then your respective treatment or the combination of treatments. And from this, we can then move from the exposure to a disease context, understanding how disease progression might affect drug distribution, but then, of course, how the drug affects the disease. So talking about drug effect and developing models of how we can relate the detailed exposure or the drug dose to a detailed assessment of the organs or the site of action exposure, and from this then derive the drug effect, often leveraging translational models, right? So using in vitro models of drug effect or animal models of drug effect, and using then these digital twins or simulation environments to translate or try to bridge between what we observe in these surrogate systems, so cells or animals, to infer about human outcome. And the very same thing we would also do in veterinary medicine, so drug development for animals. And within the chemical risk assessment, more or less the exact same solutions or approaches can be used. The only difference is that the chemicals slightly behave technically a bit different. So drugs or the chemicals used in the pharma industry are often designed to be well behaved in the context of absorption and then distribution. This is not necessarily given for the chemicals we have in industry, so industrial chemicals. So that's the challenge, to then also make sure that the software and simulation tools we use are applicable for these chemicals. But then we're also looking at exposure in a similar fashion, organ level exposure, and then not at therapeutic effect, but then adverse effect. So assessing the risk of damage, mostly damage to tissue or to processes, or for example, developmental processes during pregnancy, a very important part, or neural toxicants that can affect and also cognitive functions. And yeah, I don't want to go too much into the software related part. That will more or less be implicitly mentioned during my presentation. But underlying these services is, of course, then the technology, the software, the tools that we are building. So we need models. We're building model libraries, right? We're building knowledge bases over time that we want to reuse. So we don't have to reinvent the wheel every time. We want to build a knowledge repository that we can live off, and ideally then also over time qualify and add credibility to this knowledge repository. So we get a fully qualified ecosystem that we can use in drug development or risk assessment. And so here we're building knowledge-based simulation platforms around, chemicals, small molecules, but also novel modalities like biologics, gene therapy, to understand how these compounds distribute within the body. So we put all this knowledge into the simulation framework that is generically built for these compounds. We put physics-based understanding of how these molecules penetrate tissue, how they might penetrate into the tissue, and then bind to certain targets to affect their effect. And this knowledge is more or less generically reusable across different molecules, but then of course informed and parameterized by the individual molecule properties, right? So we're relating then molecule properties to how they will behave in the human system through these generic model frameworks. And then we can expand these model frameworks into specific or more specialized scenarios. So really adding additional knowledge and expanding this toolbox. So we've been working in radiopharmacology, where you then have to also handle different type of data, whole body radio pharmacological data, and assess this based on imaging data, for example, and translating that into an understanding of exposure. you're building models that specifically address the physiology of, for example, pregnant women, or then also related to specific women-based physiology, as often, pharmacological understanding and testing is focused around male physiology, unfortunately. So the female reproductive tract and also then risk of exposure in these tissues is often underrepresented in research, and we're building platforms in that space as well. If you move to the other side, so in very early research, so looking at screening and high throughput kind of frameworks to assess drug exposure or properties very early on, and screening for these. And then something very specific that's been coming up in recent years is the digital twins for organ on chip system, and then using these to translate into human physiology to better understand the data we get out of these novel in vitro systems. And then everything around the disease, we call them QSP disease platforms. So understanding the different pathologies, for example, diabetes, and building knowledge platforms around these diseases to then more efficiently assess drug effect. And so the physiological models that we build are really knowledge bases. So we integrate knowledge of animal species, of the whole physiology of mammalian organs. So basically understanding the similarity between the lab animals and humans, and then using that understanding of difference in our translational research. And so these platforms kind of get fed with all the data and knowledge that we gather along the drug development cycle. So just very briefly, so what we're working with is an open source tool called Open Systems Pharmacology, with the tools MOBI and PKSim for this kinetics and effect modeling. It integrates with R and a number of course, import, exporting, and reporting functionalities. So if you're interested, you can dig deeper here on opensystempspharmacology.org to understand what the software is made of. It's managed currently by a nonprofit type of all in-kind contributed consortium from industry and academia. And the community kind of lives there on GitHub. We have a user forum with quite active exchange. We're trying to foster an open collaborative environment for pushing forward systems pharmacology approaches in industry through this open pre-competitive initiative. There's a number of focus groups in the application areas of this tool that people are free to join if they're interested. And now, very importantly, what this community is also or is driving is the actual qualification. So we've established a software framework that allows us to automatically requalify intended use scenarios that are needed for regulatory submission for the software. So imagine you're assessing drug-drug interaction on an enzyme. So we've built publicly known use cases for drug interactions for which we then have built the associated template models, set up the clinical trial simulation scenarios for which these DDIs or drug interactions have been reported. And this example intended use scenario is then set up in such a way for the software that the software can automatically rebuild the scenario itself without user input. And so when we have a new software release, the software automatically rebuilds these scenarios, resimulates them, and creates the respective qualification reports. So basically, documentation of validity or quality that these models can predict the DDI outcome that was observed in the clinical. And this is fully automated. And everything that we do here is fully open source and transparent. So of course, also in the context of gaining or building credibility for this approach. Yeah, the software is accepted by regulators. We have this listed on the FDA's electronic file submission whitelist, and has been used since years also in regulatory submissions. But the point is that what we allow with this tool is a fully customizable model for drug PK and PD, which also brings, of course, some challenges in terms of the qualification. So the qualification that I showed now, or showed before, was tailored towards kinetics models, so the PBPK models only. as these are built within PKSim in a quite controlled and less flexible modeling environment. So basically what you do there is you build predefined PBPK model structures, and you then only parameterize them by your drug properties and your refined organ properties. But the whole model structure is already given based on this generic knowledge about, physiology and how drug properties interact with this physiology. But then with the Mobi tool, what we can do is we can fully customize these models. And once we go into customization, of course, quality control becomes a challenge. And that means qualification and, retaining that credibility of the model and the modeling framework is a challenge. So I want to outline here a bit how such customized platforms or model platforms that then go into the disease modeling space, where we combine drug kinetics with drug effect and disease progression, can look like. And so you can better imagine what the challenges related to this might be. And we're working quite close with this open source community, as we also provide the software developers that develop this tool and have been quite instrumental in achieving the framework that's needed to qualify such customized platforms. Yes, so just maybe I should have brought this a bit earlier, but so this is more or less the context of knowledge that's contained in these models, right? So we're building kinetic compartmental models across the different scales. So going from the cellular compartment over organs, over then the whole body physiology, then also parameterizing this for populations, similarly to what Stephen earlier showed for his medical device models. Yeah, I guess I don't have to motivate necessarily this modeling approach, but so given that we are getting more complex modalities in terms of the biologic space, novel modalities, but also, of course, the space of where we can search for novel therapies, druggability of targets, and so forth. Also, the related biology becomes more and more complex. So really understanding translatability between the test systems from in vitro to animal to human is a challenge. And also this has been leading, of course, this challenge of, that your gut feeling might be enough to make the next decision in your development cycle, has led to an increase in drug attrition and failures. So that this mechanistic modeling approach that's intended to increase your understanding of what's actually happening with your drug within the system, as opposed to kind of resolve this issue to get a better understanding of how findings in earlier development are translatable to the clinic. These QSP-related approaches have also found their way into also regulatory submissions. So supporting such decisions or rationales in study design and so forth. Yeah, what we do get quite a bit is also then drug effect related assessments, of course, of first in human studies. Also pediatric scalings, for example, if you also want to assess the drug effect scalability across age and so forth. So increasingly also applied in regulatory applications. But of course, given the context of use, this is also understandable, as these models can more or less be applied across all stages of drug development. And yeah, just very briefly, of course, to assess proof of biology, we are contextualizing the target kinetics and the whole physiology of the test organism. We're contextualizing drug properties to drug PK, so understanding the pharmacokinetic behavior across species, so then predicting for the first in human study, and then also using that knowledge to optimize study design. Given the generic approach based on molecular properties or compound properties, we have an easy time doing benchmarking against candidate drugs or competitor drugs, and then leveraging disease knowledge, population knowledge, physiological knowledge. We scale across age, children, geriatric age groups, or across disease population. So that really we can go throughout the different clinical designs, right? And this is really leveraged or enabled through the physiologically based and yeah, mechanistic type of model context that really contextualizes this knowledge as best possible, but also as efficiently as possible, so that we can still handle the model complexity. Okay, I'll skip this one. But as we move then, of course, into larger type of use context, right? We're expanding these models. So, for example, how does it look in pregnancy? We're adding a fetus physiology into these models. Then on top, we add potentially some disease. So what's shown here is, for example, an endocrine system and how this could be disrupted by chemicals or by compounds. So adding complex also drug interactions or endogenous type of regulatory systems into these physiological models to then assess either disease progression or just a system that might be affected by a chemical or a drug. Or we even built these into multi-drug type of platforms where you have multiple compounds in development for an indication, so that these models can become quite large. We've built also, for example, the ocular absorption structures. Or as mentioned earlier, also different platforms related to women's health that can quite significantly increase the size of such a model. And this size and adaptability, of course, makes it quite challenging. I'll skip this to certify or qualify such a platform. If you know the software or not, I just want to give you a very, very brief intro about the logic, so that you know a bit about what I'm now presenting as a solution to this challenge. So the way these tools are built is they're kind of built by building blocks. So what you're doing in PKSim, you have a library where you can load individuals from a predefined population of a certain ethnicity. You can then define your compound and your compound properties and potentially an administration protocol. You might have a formulation for an oral compound, or you can even add expression profiles of enzymes, transporters, target proteins that are involved. And these are all database driven, so you can create your PBPK model. But that makes it quite easy for someone who might not be an expert modeler to build a PBK model. If you then want to customize it, these whole models are then translated into a more technical context. So, but more or less in the back end, also these PKSim physiological models look the same. So basically, the organism is in principle a container structure, right, with intracellular space, interstitial space of organs, of blood, plasma space, blood cell space. And between these spaces or compartments, there can be processes like passive transports of diffusion or active transports like enzyme or transporter related processes. Then, for example, also transformative steps are then not enzymes or compound-related enzymatic reactions, but we call them just reactions, and this can be any type of reactions or a signaling network in the effect modeling space. So, and now what we have more or less established is that within the software, in the past, what was done is to customize PBK models. Your extensions were more or less hard-coded into the PBK model, which allowed us not to more or less flexibly work with the model after everything had been hard-coded into the same model structure. So we were no longer able to look at components independently and potentially also improve these components independently. So we've moved into a modularization logic now, so that more or less what we do is if we take these PBK models into the MOBI tool, where we can then customize it. These customizations are developed as their own modules. So basically, you then have modules of the original kinetic of a compound, and then modules of whatever you want to add on top of this kinetic. So an extra organ, a signaling cascade, a disease progression model that affects the physiology. So these extra modules are developed separately, and then the software gives you the logic to combine these as needed in a specific intended use scenario to then set up a simulation. And yeah, I'm also going to skip this. So it's more or less the last technical slide. I want to give you this very busy slide. So the workflow would then be as follows: to build or manage such an model-informed drug development platform, so that we can call it in the end more or less like an ecosystem, or the backbone, the digital twin backbone of drug R&D. So you can imagine you can build then over time a library of compounds for kinetic models, so this PBPK library, and you can also build a library of such modules that you add on top. So you can add, you can define a tumor that you want to reuse in all sorts of models, or an effect model that you might want to reuse. And then you can combine these into such a platform that connects kinetics and disease. But as these modules still are separable, your other teams might be able to continue working on the PBPK model because they don't need your drug effect or disease model in the context of formulation development, drug-drug interaction, and so forth. And as they evolve and refine this PBPK model, they can provide it back to the team that works on the integrated disease platform to update this model separately in this platform. Yeah, thereby always have an up-to-date model version. And given the modularity, this automated requalification routine can also handle now a customized model platform, as the program understands what modules relate to the original PKSim model, and there we already have the auto assembly of models established. And adding these modules then, and the logic is then an extension, a logical extension to this automated model reassembly and qualification workflow, so that we can in the end have a bench to, let's say, bedside and back kind of modeling environment that allows continuous maintenance of these models across functions and timelines within the drug development. Yeah. And we've had a number of use cases in this space, so working well on ocular formulations, and then expanding the default structure. So an ocular absorption model is not part of PKSim. We've developed this as a module so that we can flexibly add this to the software. And of course, as it's generic and could be applied to all sorts of modalities, and look into ocular distribution and absorption. A diabetes platform, which reflects most modalities that are used, for example, in diabetes. So in the past, all of these PK models of all those compounds always glued together can now be use case specific selected. So of course, you might not always dose all of these compounds in one scenario, but maybe two or three at the most. And then also looking at different populations separately, like type 1 diabetes or type 2 diabetes. And potentially for some compounds, you might need a more detailed physiology. For example, for SGLT2 inhibitors, you need a more detailed renal physiological representation to really also assess this drug transporter's inhibition effect on the blood glucose kinetics, for example. Yeah. Some more examples. So, for example, also here for adverse effect, we have the thyroid disruptors platform, where we have the thyroid hormone endocrine regulatory system implemented within the platform as a module. And then we can evaluate this with a number of chemicals and assess the risk of these chemicals to disturb this endocrine system. And this is implemented, for example, then for rat and humans, so that we have a way to then translationally assess different drugs across the two species. Yes. Similarly, then if we move into the novel modality space, so what we've then also used or done is to expand the base distribution model with different aspects that are needed for the different novel modalities that we have these days. So moving from a traditional monoclonal antibody to bispecific antibodies, where we also then have to assess potentially immunological cells, or like T cell kinetics and distribution across the system. So expanding then a system with a T cell migration and distribution model to be able to interact then with the compound. Yes, or immune cell or immune function related modules that we can then add on top such a platform, or specific kinetics related to, for example, gene therapy and their delivery into the system. All right, so then potentially adding modules on top of the default monoclonal antibody distribution model, then also related to, for example, target-mediated clearance and drug disposition. We can work with either tissue-related receptors or soluble receptors and targets, and then, of course, add different types of PD and physiology that might be relevant for these modalities. Yeah, so I think we don't have too much time left. So yeah, I want to more or less stop here to not steal too much time from the last speaker. So yeah, in principle, the congruent aspects can be done in talks as well, and these slides have just outlined that as well for talks-related effect systems. So thanks everyone, and with this, I want to hand it over to the next speaker, Philippe.
Thank you. Just share my screen. All right. Stephan, can you please confirm that you see my screen? Or somebody else?
Yeah, I can confirm.
Yes.
So.
All right, excellent. Thank you. All right, thanks a lot. My name is Philippe Favre. I work for Zimmer Biomet, and I would like to show you today, first of all, talk a little bit about the company so it's clear what we do, but the focus will be really on the use of modeling in the orthopedics industry in general. First, I will focus on the classic use of modeling, and by classic, I mean the widely accepted use of modeling within regulatory submissions. This includes finite element analysis in a wide range of simulation types and physics, and also anatomical analysis. And then I will talk about an emerging application of modeling, which really goes in the direction of what Steve has shown, In-Silico clinical trials. Right, first of all, a few words about me. So I'm married, I have 2 boys. I like running, skiing, and traveling. In my studies, I'm actually a mechanical engineer, and the first job I kept for 8 years. I was working in the orthopedics hospital at the university in Zurich. And there I was working every day with surgeons, trying to address their questions on biomechanics using cadaver testing, cell bones, or modeling. Did my PhD at DETH on shoulder modeling, and in parallel, I was also leading the clinical biomechanics group at DETH. And then in 2011, I moved to the industry, to Zimmer Biomet, and I've been in the computational biomechanics since then, a group that I now lead. So Zimmer Biomet, it's an American company based in Warsaw, Indiana, has roughly 17,000 team members spread around the world. We have manufacturing facilities in America, Europe, and Asia, and it's a Fortune 500 company, and with sales of 7.6 billion last year. All right, the products we offer are basically almost anything that you could think of for musculoskeletal issues that people might have. It goes from the large joints such as hip, knees, shoulder, but also thoracic, trauma. And we're also, since a few years, really putting a lot of emphasis in robotics and data. All right, that was for the small pitch about the company. Now, the topic that interests us today is modeling. And here I would like to show you really the models that are currently used, that are widely accepted in regulatory submissions. So as a company, we're responsible to establish the product safety and efficacy before we go to the market. And there are several ways to show that our products are safe. We do that through preclinical testing before the product is launched, but also with clinical follow-up and post-market surveillance. So making sure that the product, once implanted in the patients, works well. And the classic use of modeling at our company, but I would say in general in the industry, is in the preclinical testing phase. So before the product is launched in the market. Here are the typical risks that we investigate. So on the left, you see the clinical issues that might arise with implants in general. So one typical is implant fracture. And for those clinical issues, we have an in-silico equivalent. So typically for implant fracture, we could look at, in the models, at implant fatigue stress. For septic loosening, which is loosening of the implant in the bone, we could look at micromotion and interface strain. So we have a lot of different metrics we can use to try to replicate what might happen in the clinics. And this is really our day-to-day work. One of the main use of simulation is for worst case identification. And one of the main tools, obviously, is finite element analysis. So what I'm showing here is a shoulder system. And you see there are a lot of components because people have different conditions, different issues that the surgeon need to address. And all those components are also available in different sizes. In this study here, we were interested to identify the worst case stem, so the component that is at the bottom of that image. And obviously, there's not an easy answer, especially when you consider all those components in different sizes. So you don't want to test those on the machine, obviously. One fatigue test takes 2 weeks. So what we've done here, and what we do very often, is use finite element modeling. In this case, we ran over 100 models, which finally indicated which conditions, which load case, which component assemblies lead to the worst case for that whole system. And then with that information, we just test that one and know that the system then will pass. So obviously with modeling, there is significant time and financial gain as well. Another thing that we use modeling for is for design optimization. Here is an example where you actually see a liner that is assembled to a metallic component. And the liner has to be assembled by the surgeon during the operation. And once assembled, it has to hold as long as the patient will live, hopefully. And so here we had an assembly force target that was as low as possible. Basically, we want the surgeon to be able to assemble that component easily, but once in the patient, we want that system to hold as much as possible. So we had some targets here showed in dashed lines, and you see the different design iterations on the horizontal axis. And you see here it was done in a very, I would say, conventional manner, where the engineer was coming up with a design, would run the FEA and see where it lands, and then, well, maybe I try this and update the design and see where it goes. But at some point, after 11 design iterations, they realized that, well, they were not bad on lever out, but the assembly force was still way too high. That's where actually we decided to incorporate design of experiments. So telling the computer to modify some of the dimensions and find the optimal. And here in 3 design optimizations, both acceptance criteria were met. So I think it's a really nice example to show that, using conventional methods, using very smart engineers, trying to come up with ideas is good, but you can really have a much better outcome if you use statistical methods. Another way why, or another aspect where modeling is really great is for enriching physical testing. As I said before, one of the key things we're interested in, or a key risk, is aseptic loosening. And that is when the implant moves too much with respect to the host bone. That is something typically that you could test in a benchtop test using a cadaver. This is an example of an implant. And to test that physically, we created a small window in the bone and looked at the interface between the bone and the implant when the implant was loaded. So hopefully you can see the metallic component on the top of that image moving with respect to the bone as it's loaded. Then we used digital image correlation, which allows us to actually quantify that motion. But that is only in that very, very small local location. Now, what we did is we created an FEA model of that exact same bone, creating the same windows as were done in the physical test, and looked at the motion of the nodes from the implant and the bone under the same load. And what that allows us is eventually, instead of just having the information at that very small location, once we validated the model against that experiment, it allows us to look at the full surface of the implant. So we get a full micromotion distribution. We get all components of the micromotion, because that's a 3D metric. And what we've done also is, instead of just running one load case, which with cadaver you can do really more, here with the FEA, we were able to run a lot of different clinically relevant load cases. And so this is really impossible at the moment to do with modeling, without modeling, sorry. So a lot of value to having those models. Away from the FEA, now we look at anatomical studies. So those are purely geometric analysis. If you have a database of models that would come from CT scans of patients, and you have the CAD models of your implants, you can, on the computer, do a virtual surgery. So you can place the component where it should be and analyze different things. And what that is really helpful for is to make sure that your design has the right shape, that it will fit the bone, but also it will fit the population so that you have the right sizes, basically. And so an example where the type of simulation was used was for one of our key knee systems, sorry. And what was done here is taking the tibia and cutting it as would be done in the surgery, but on the computer. So we took the database of bones, cut them as the surgeon would do on the computer, and also included some variability. So cutting a little bit too deep or a little less deep at an angle, etc., as would occur in the surgery. And this gave us all those little contours here that you would get if you cut those bones in orange. That helped us then define what the ideal boundary of the implant would be, which is now the blue contour, which allows, or which would prevent overhang of the plateau on the bone, which is something that you want to avoid at all costs. And that was actually the first design that was asymmetric. And that helped us then also compare with different designs. Having that asymmetry doesn't require the surgeon to rotate the component to try to fit the implant to the bone. That is asymmetric. And so that has a really wide range of implications, notably that you can reproduce your axis of motion really accurately. And that would be obviously very impractical to do with modeling. You would need a large number of cadavers, you would have to image them, etc., etc. Another thing that you can do with those anatomical analysis is also look at motion. Obviously, when you construct a joint, you want to have a good enough motion, large enough motion, and that is also something that you can do on the computer. You can move around the center of rotation and identify when you would get contact between the bone and the implant. So those were the classic uses that we have in my company. Those are the things that we submit as well to regulatory agencies in the world. And a very important aspect that has already been very well explained is the model credibility. So this is an absolutely central aspect of the models we do. Every model study that we submit has credibility assessment according to the SMAB/NB40 standard. And we do that for all studies comparing to a benchtop or an in vitro test. Now, the emerging application is the In-Silico clinical trials. So this is something we've been pushing in the last years because that's a technology we really believe in at Zimmer Biomet. And I would like to show you a little bit what we've done. So again, the classic use of modeling is preclinical testing. What we're interested in, though, is to start modeling what happens in the patient. So that really goes in a similar direction to what Steve has shown with the Living Heart project. And the concept of in-silico clinical trials, or at least the way we wanted to use it, is if you have a number of real patients for your clinical trial, the ideal case would be to actually cover all the demographics, all the use conditions, all the morphologies and surgeries that you have in the population. Because that's what you want to study in a clinical trial. You want to see how your device is performing in men, women, old people, young people, etc. But because the clinical trials are limited in terms of number of patients, inevitably you're going to miss some of those cases. So the concept here of in-silico clinical trials is to fill those gaps with virtual patients. So we really have models, computer models that would fill those gaps. Now, if you have a pipeline that can fill those gaps, there is nothing except for computational time that prevents you to actually generate a lot more virtual patients. So another benefit is not just fill those gaps, but you can also really increase the statistical power of your study. And also, you could also look at very specific conditions, very extreme conditions that you would very rarely get in the clinical trial. So you get to learn a lot more about your system. So lots of benefits, really. How that works technically, very quickly. You start from a CT scan and you segment the bone so that you get a 3D geometry for that patient. Again, you can perform virtual surgery on the computer, placing the component as a surgeon would do. Then you move to the FEA world where you mesh your geometry. You can assign the material properties of the bone from the CT scan information. You can apply loading based on musculoskeletal models. And you can look at any kind of metric of interest. So you could do this, that pipeline, you can do that for one bone. Now, what we're interested in is generating a population. So of course, you can do this for a few more bones. So that's where you introduce patient variability, which is a key parameter in the clinical study, right? So patient variability comes from different anatomies. So you can do that for several patients. You can also play with the bone density, showing what would happen in the same bone with good density or poor bone. You can also play with load, showing you what would happen if the patient does different activities. Another very important source of variability, which is captured in a clinical trial, also comes from the surgical variability. And that is something we can also do in the model by looking at different component combinations, different sizing schemes, or different stem or component alignments, as the surgeon would do. So what we've done here for that study is we started with 45 unique anatomies. So 45 scans from 45 different patients. Then as I just explained, we can introduce a lot of surgical variability. So we looked at 2 different devices. We looked at different stem length, different tilting of the implant, different sizing schemes of the implant. And doing this for all of those 45 bones actually increased the final number of plausible interventions to over 500. And that is interesting because 500 is roughly close to 10 times the size of a clinical trial, a typical trial in orthopedics. So it just shows the statistical power increase. And then also we looked at 3 different load cases for those 500 models on those 2 different systems, and looking at 2 different outcomes of interest, so humeral loosening and stress shielding, which in the end gave us 3,000 solved models. So really a large number. So that was the technical thing, technical aspects along that journey. That was one component. There were many other things that were actually that had to be developed along the way. So all of those steps are actually new, and I will go through a few of them to let you know what we've done here. One thing that we've done as well is on any of those new aspects, we published our approach. This was done just to make sure that it is clear to everyone what we do, and also for us to get an endorsement from the scientific community to show that, yes, what we are doing is also scientifically sound. And that is something that is really important in a regulatory environment. Another aspect that I would like to show is model validation. So the ASM V40 is absolutely great, and we use that every day for benchtop tests or in vitro tests validation. This is absolutely great for well-controlled and characterized data. Now, if you have less well-controlled and characterized data and you have more in vivo variability, that standard is less appropriate. And that was our problem here when we tried to model a clinical trial where we have a lot of in vivo variability. And basically, the data that we get is absolutely not controlled. We just get the patient we get. So we had a lot of discussions, and actually we decided to develop our own methods. So everything that Stephen showed in the first talk pertaining to the FDA guidance and how to model, how to validate the models against clinical data, that was not available at the time of our study. So keep that in mind. So what we proposed at the time was to have 2 aspects. One, benchtop validation, and the second, the clinical validation. So the benchtop validation, we followed ASM V40 completely, and this was done to ensure the physics are modeled correctly. Now, the step we added was to add a clinical comparator. And this was done to ensure that we predict the clinical performance of the system in a population, and that we can reproduce significant differentiations that were observed in the clinics. So the example here for stress shielding, how we validated this. So we had a step, again, following the ASM V40, comparing to benchtop testing. Now for the clinical validation, we found a published study that showed that larger implants, so when the surgeon sizes the implant a little larger than what he could or she could, there's a higher risk of stress shielding on that side of the bone. We chose that study because it was well powered, it was well designed. They were seeing significant differences between things that we could reproduce in a model. But this was not in isolated cases, so that the stress shielding that is observed here with the difference in sizing is something that has been confirmed in many other studies. So in order to reproduce that study, we took 35 bones. We used the stem that was actually used in that very same clinical trial or clinical study. and did a lot of sensitivity analysis. So the mesh sensitivity, obviously, but also looked at the stem size, the stem alignment, the loading, the material property influence, to really characterize the model and understand what the important parameters are. Then looking at over a pretty large number of virtual bones, we were able to see the same significant differences between the two groups with oversized and properly sized stems, as you can see in the graph. Another thing that we were able to see is a change in strain energy density, which is the metric to look at bone resorption, which was also seen here, actually in the same location as was seen in the clinical trial. So this doesn't stop there. This is really just the comparison of the two, of the model and the comparator. We also proposed a very similar approach to the SME-VNV40, looking at all the different credibility factors, both on the model and on the clinical trial. And we adapted the different factors and how they're assessed. So I don't want to go through that table that is published. If you want to see it, please check the paper. But one example here is on the computational model and inputs. A low credibility would be to have only one representative configuration. Have maybe several bones, but only one way to implant the bone, implant the prosthesis perfectly, for example. Whereas high would be a lot more bones with sensitivity on the surgical variability, for example. Another example is the applicability. So how the COU model, how good the COU model and the validation model are similar. So low would be, it's a similar device. For example, if we used the study that I've just shown for another shoulder implant, well, the study would have that credibility factor would be low. If it was exactly on that same device, it would be high. So this is an example of how we also translated the SME-VNV credibility factors to a clinical application. Another step I would like to discuss briefly is a regulatory submission. So we've done all that work to actually support a regulatory submission. On the regulatory side, the InSilico clinical trial has been supported by different initiatives, both from the FDA as well as on the EU side, which has been funding the Avicenna Alliance, for example. And so here you see that, yeah, there are a lot of guidances, lots of documents that really helped us build confidence on the InSilico clinical trials as far as the regulatory submission. We had, of course, we want to increase the chances of acceptance by the regulator of such data. And so what we propose here also to make sure that the regulator is going to look at the data in a good way is follow any guidelines that is related to what you do. For example, the guidelines on clinical trials, if you do this numerically, it's good to follow the same things. As Stephen also pointed out, there are a lot of guidances now on reporting computational studies, on validated models, and that is really key to follow. We've also had open and regular communications with the regulators to make sure that the way we model our ISCT or how we develop our models are aligned with their view. And again, we publish the approach as much as possible to show that what we do is acceptable scientifically and is sound. All right, with this, I have a conclusion slide. That's my last one. So basically, computer modeling is heavily leveraged. At our company, some other companies may use less, some more, but that's our example. I think where it is mostly powerful is for worst case identification, because we can reduce the physical testing by a lot. It also allows us to have better implants. I've shown you the example where we optimize the design, and that allows us to be shorter and faster to the market. V&V is absolutely essential. It's a lot of work, of course, but it really helped us build the credibility in everything we do. So that's should not be underestimated. It needs to be done well and takes time, but that's really priceless. And ISCT, the example I showed you as an emerging application, of course, it's emerging, so it's still starting, and we need a lot more standardization and acceptance of the technology here. All right, with this, I'm done. So as Stephen said, I'm also happy to take questions offline if you want to get in touch with me.
All right. Thanks, guys. I don't know. Laura, can you let us know if there's any questions in the chat or anything? Does not look like it.
Sorry, I can't see any questions so far.
Okay. Perfect. Got a couple more minutes if anybody has any, or anybody else wants to. I think we're good then.
One.
Oh, there we go. Failures. There's no such thing as failures. Case studies of failures. I will say from our perspective, we don't have any published failures. Typically for us, we're behind confidentiality in that circumstance. Unsuccessfully within a submission. Why? Oh, plenty of those. Philippe or Stephan, do you want to comment on any of those? And I'll chime in as well. Jeff asked if there were any unsuccessful submissions using modeling and simulation.
Well, I mean, in most cases, you're trying to argue for, a certain beneficial outcome, or, you're saving yourself a certain extra, clinical trial. And it kind of depends on how well your argumentation is lined up with your modeling exercise to see if you can convince the reviewer or not, right? So it's often also a human factor, both on the side of the modeler or the side of the reviewer. And yes, I mean, sometimes you have argumentations in which initially you get a dispute and then you're successful after discussion, or you might not get success. But these are then usually boundary condition type of applications that are not clearly defined, or where you kind of started off with already a borderline type of, knowledge base, and you were trying to make maybe a case with sparse data. Trying can't hurt, as you might say. But yeah, usually for well-defined applications, no failures, no.
That was good. Philippe, anything you want to add?
No, I totally agree. I think along the way we know exactly, what we're allowed to do and the limitations of what we can do. We do get questions, and we answer them, and in general it goes well. We do see differences depending on the countries as well. So some countries will ask more questions than others.
Yeah,
but I wouldn't say that we had failures.
Yeah, and I'll say too, oftentimes the failure or the response from the regulatory agency is a motivation to do some additional testing or some other work to address whatever the question is. Oftentimes we'll be doing a fatigue analysis, but they'll ask relevant questions about fretting or corrosion or wear, and then it's a matter of looking back at the analysis and comparing that to the testing and maybe running some additional testing to address whatever those questions are. Most circumstances, we've been given the opportunity to respond in a technical way, which with modeling simulation is really nice because you can go back and you can look at what was done and you can think about that and review those results.
Can I ask a question to both Stephan and Philippe and yourself, Stephen. Do you have a sense of how often a typical regulatory file contains modeling and simulation evidence, if not as the pivotal evidence, which I suspect is very little, but at least sort of additional evidence? And I'd like to see if it's possible to have a sense of that in pharma and in devices separately.
Sure. Yeah, I'll say just on the device side, and I won't speak for ortho because I think Philippe probably has a better handle on that. But from the cardio side of things, I would say just about every application has some level of modeling and simulation. Typically there, and if you follow the standards, it's actually called for and directed that you need to do it in order to justify certain conditions that you're going to test against. So at least in the cardio space, it is in just about every, if not every application for the typical use cases. It's a bit lower number, of course, for the non-traditional applications. But for the just traditional PMA applications, so for the class 3 implantable life-saving devices, it's in everyone.
So in the ortho, we're not at that level yet. I think there are cases, when you develop a device that's close enough to the predicate, where it's pretty clear what the worst case is, and the design doesn't need a crazy amount of optimization maybe. And so I wouldn't say in all of them, but more like really where you don't know the worst case, it's not obvious, you cannot rationalize, and that's where we come in. But it's not all cases.
Yeah, in the pharma space, of course, it depends which submissions, right? You have multiple along the way, and then also what type of modalities, so the chemicals or the novel modalities. I would say in a larger pharma company, most submissions have a modeling component, but there it also then depends what type of modeling, right? So we have the more simpler, I don't know, statistical models, non-compartmented analysis, then towards simpler models to the models I presented, which are quite complex, and these are yeah, still the exception, I would say, in submissions. So as you saw in this graph where, the numbers were basically 0 just 10 years ago. Yeah, so it depends. So it varies across, but I would say, if I would say a ballpark gut feeling, it's 50%. But so it's a spectrum. It's like a heat map, I'd say.
I think you have another question probably for Stephan again. From Deepanjan. So he was saying population modeling, virtual bioequivalence, regulatory acceptance of prediction errors, and when geometric mean tends to get lower than arithmetic mean, how to handle such submissions successfully.
Well, that's a quite specific
question.
I don't think there's a golden rule to that. I think it depends on the use case, why the difference between arithmetic and geometric standard deviation is observed in terms of sample size and so forth, and outliers. So I don't think that's easily answered.
Let's keep all those tough questions on the pharma side, please.
Very good. So perhaps thinking about the time, we should be bringing the session to an end. But it is, thanks very much to all three of you for sharing your expertise and your experience as well. And just for everyone else, we will have a number of additional sessions and webinars over the next 4 to 6 weeks going on. And then we will start, as Stephan was intimating, a sequence of case studies. So we're going to get into a lot more detail into each of those. And yes, Stephan, that was precisely what we want to do, is go through the standards and pick a bit like we did today, but rather than having 3 in the 2 hours, have a lot more detail to go in depth. And there will be a team behind each of them. So if anybody is interested in being involved and you haven't sort of connected with us, just drop, Lara and myself an email, and we'll see whether we can slot you into any of the existing case studies that we have already planned. And with that, I'll pass to Lara, who will remind us what the next webinar is going to be.
Thank you. I'll just share my screen.
Yeah, I guess we'll also be happy to take questions offline at any time.
So if everyone can see my screen, the next webinar will be on this Friday at a different time from 2 till 4 GMT. This will be a webinar with some folks from regulatory discussing the promise and requirements of model-informed computational evidence as a way to transform regulatory pathways. And you can sign up for this via Eventbrite.
Thank you, Lara. This is going to be a very exciting event. We have three speakers from MHRA lineup, and they will provide both the perspective from both pharma and devices. And it's an opportunity to also put questions. Although, I must say that they are in the process of starting to take on board this sort of evidence. So they're also participating to learn and engage with the community. So it's going to be a great opportunity, I think. I think there is a question, if I'm correct, Jimmy, Oh, God. So probably we'll have to leave it there. But thanks to everyone for your participation, and we'll be in touch. Thanks again. Have a great day.
Everybody.